And so we have here with us today, Lonneke Scheffer and Milena Pavlovic. I want you guys to give a thumbnail sketch of sort of your bios and where you are right now. And then we're going to talk a little bit about the paper and what's going on with ImmuneML. So why don't you start, Lonneke?
Yeah, so hello, my name is Lonneke Scheffer. I'm a PhD student in computational immunology at the University of Oslo. So my background is a bit in bioinformatics, and during my PhD, I'm working on applying machine learning to adaptive immune receptor repertoire data, which is I think with my bio, I kind of covered pretty much Milena's bio as well for the large part.
So you are both doing, you're working in the same laboratory, is that correct?
Yes, that is correct. Yeah, and we collaborated together on this software platform, ImmuneML.
Fantastic. And let's have a little word from Milena, a little bit about your background.
Yeah, so as Lonneke said, I'm a PhD student at the University of Oslo, the Department of Informatics. And the main focus of my PhD is machine learning and its application to diagnostics and antigen binding prediction in computational immunology. So pretty much the main use cases that the ImmuneML platform covers.
Fantastic. And so let's talk about the paper. The paper was published in Nature Machine Intelligence, November 16th, 2021. The ImmuneML ecosystem for machine learning analysis of adaptive immune receptor repertoires. So let's kind of distill that. You know, what are you really, what are you presenting there? I mean, you're talking about these AIRRs, adaptive immune receptor repertoires. Those are the key targets for biomedical research, right? Because they're kind of like recording past and ongoing adaptive immune responses. I'm basically just reading your abstract. But, you know, so that's important, but it's really like the layer that you're adding is this, you know, machine learning ability, or you're training the machine learning, right, to identify the differences in these patterns. Is that correct?
Yeah, so our adaptive immune system, so this adaptive immune repertoire, it's basically a collection of a lot of tiny receptors. And so one way that you can do disease diagnostics is by looking at which receptors are present in the immune repertoire. And we are using machine learning to try to identify which receptors would be relevant for a given disease of interest. So that could be, for example, type 1 diabetes. So these, it would be about identifying which receptors would be relevant or what kind of patterns within the DNA or protein sequences of these immune receptors are relevant to kind of predict this relevance for the disease.
Right.
And this ImmuneML platform is kind of one platform where different machine learning methods that can be applied to this AIR data come together and can be explored by the user.
You are also offering this up to the community, right, in an open science, you know, manner, which is, you know, really excellent. But so if we want to talk about diabetes, also type 1 diabetes specifically, right? There's this whole prodrome where people are that, you know, they have many of them have, or most of them have a genetic predisposition or risk. And then, you know, some of them will present an autoantibody, and then maybe the second autoantibody, and then maybe the third, and then diagnosis. And so there's this prodrome, right? The lead up to the disease. So using, like, can you imagine using your system to kind of investigate that prodrome, right? Like what's happening at during the different windows. And you can imagine that this would be a really powerful tool, because it could, you know, allow understanding of the windows of opportunity for clinical intervention, or sort of just really just understanding the etiology of the disease, like what is happening, because that's still unknown.
Yeah, so one thing that's probably good to mention is that ImmuneML is kind of a general framework. So it's not necessarily, it can be applied to type 1D, but it can be, it's more like specific to this air data and not necessarily for one particular disease. But we do offer some functionalities related to giving a bit more insight into what the machine learning methods actually learn, instead of just having machine learning methods that just have a good prediction accuracy, to actually get a bit of insight in, hey, what were the patterns that might have been relevant in this disease, and what is it actually that we are making our predictions based on. I don't know if Milena has something to add as well to this.
Yeah.
Yeah, maybe just one minor comment related to different like stages of the disease. So one thing that for the platform, all the immune repertoires are the same, and then we try to find some sort of differences between them that are kind of significant in terms of, for example, diseased people or people who do not have a disease. But another class of people could be, for example, people who are relatives to the ones that have developed the disease and have similar genetic background, but do not yet have the disease, and multiple groups like that. So instead of just learning to distinguish between people who don't have any prior history of a certain disease, we could also look into distinguishing between people who have similar genetic background, but still different diseases. So that could maybe be one step towards something that you mentioned.
Yeah, no, that's an excellent point, to sort of, bin the different subgroups and examine them and compare, basically compare them. I wonder if someone approached you and they were kind of a novice, in using this, this tool. How would you counsel them? Like, how would you, so suppose they're like a cell biologist, they're, been looking at like beta cells and they're kind of interested in maybe examining the lead up to the disease. Like, how would you be able to walk them through this process? Would you provide support? Or, how would it work?
I'm kind of waiting, like, who's going to answer the question, me or Milena? So what I can say about kind of, so we have these different imagined user groups for ImmuneML, and one of those user groups would be people with more of a biology, immunology background, and not necessarily so much machine learning. We do provide very extensive Galaxy web interface for these for people who don't necessarily have so much machine learning knowledge. So with this Galaxy web interface, we basically have a more text-based, instead of writing like analysis specification where you have to type all the parameters into one big specification file. The user is kind of guided through making these decisions based on text-based questions of what kind of signal what do you think that the signal of the disease looks like? Like, I have this kind of presumption about, basically asking several kind of presumptions that people might have about their data, and then we kind of construct the more technical analysis parameters in the background and have some sensible default settings for the parameters that we don't expect that the immunologist could really sensibly choose themselves.
Because it is an interface. It's really an interface. You have an expertise in this whole machine learning side, but a lot of scientists have their own expertise and they don't always overlap. So it's fantastic that you're kind of bringing this to other scientists. And I think it's really an excellent, platform, and a really a fantastic opportunity for people to look at their data in a new way.
Thank you very much. And we've tried very hard also to kind of have easy ways to kind of step into the ImmuneML platform, have quite extensive documentation for different kind of purposes, either for the immunologist who is not so expert in machine learning and wants to, you know, have a first test run on their data, to the machine learning expert who wants to integrate their new method into the platform. So we cover the whole range of different possible users.
Fantastic. How about
If I
clinical users? You know, sort of the MD, PhD, or the MD who's interested in kind of mining some clinical data. Is that an opportunity too?
Yes.
Good.
So they can use for example, this interface that Lonneke mentioned. So through this web-based tool, and we support a lot of different data formats. So depending on the tool they use to pre-process the data, the sequencing data, like MXCR, or if they use 10x genomics for sequencing or something else, we can right away kind of import all those data types. And they can very quickly get started into some basic analysis. And we also have, in addition to that, integrated with a few of other existing tools in this field, and implemented some already existing papers that were published for diagnostics. So that is readily available to people who are getting started.
It's fantastic. The onboarding seems pretty seamless. Has it been pretty seamless when you've had to sort of, you know, show people how to engage in this?
Well, of course, there's a whole history behind the onboarding becoming seamless. It's we've gone through many different rounds of feedback to get to the point where we are, which is, I mean, I think the best feedback that you can get is just from people who have actually not used your tool and are trying to use it for the first time. And we've tried several times in our own lab to kind of have these sessions with people that have not used ImmuneML before, and kind of try to not give too much prior knowledge and see if they can figure it out purely based on the documentation. And we got in a lot of valuable feedback from that, and made some pretty big changes to the platform actually, in order to really facilitate the fact that that is actually usable. So that has been there has been quite a history of stress testing ImmuneML in that way. But
that's fantastic though. I mean, that's really how you can make something just better and better and better. When we talk, do you want to talk a little bit more, sort of dig into some of the more, you know, like highlight some of the interesting parts of the paper that you think people in the type 1 diabetes field might find useful, or maybe direct them to some sections that you think are really interesting? Anybody? I know I see one of the other authors is here too. So feel free to dive in, anyone. It's a free-flowing conversation. Maybe. I think, I like the fact that you're talking about there was this whole widespread adoption of this ARML has been inhibited by lack of reproducibility and transparency and interoperability. When you talk about interoperability, what do you mean by that?
So there's a lot of different tools developed by different research groups. And when you are in a situation like that, there is a risk that everyone kind of has their own ways of doing things and has their own standards, has their own formats, has their own meanings of what kind of the same thing means, which can be quite a challenge to navigate. I mean, for example, the fact that there are, I think in ImmuneML we support more than 10 different input formats, which all kind of have their own little ways in which maybe small things need to be changed about like the raw numbers that are in the file to actually make them mean the same thing. It's actually it can be quite a challenge, and we're trying as much as possible to support the standard. So there's this AIR standard, for example, for what data formats are supposed to look like.
Mm-hmm.
So we are trying to, as much as possible, output our own results in kind of standardized formats that should be as usable as possible in combination with other tools. But then also at the same time, we try to accommodate for all these specific tools that are out there and the particular results they output, to make it actually work together in one streamlined analysis.
Yeah, that's a big job.
And the Galaxy web interface actually also kind of comes into play again there, where, for example, we have integrated some tools for directly importing data from public databases like the iReceptor Plus database. This is another way in which we support interoperability with, in this case, this database that you can directly in one web interface. You don't need to install anything. You just go to the website. You can just directly download some relevant data, apply ImmuneML, apply maybe some other relevant tools. So to actually have everything in one place and have the user spend as little time as possible trying to navigate how should I now, change my output file to make it compatible with this next tool.
Yeah, and I mean, it sounds really seamless. And I wonder, so for instance, what if you have sort of interdisciplinary users, they all want to collaborate, and is that going to be fairly straightforward as well? What if somebody's, in Europe and somebody's in the States or Canada? Is it easy for people to access the data? I mean, I'm thinking of like how protocols.io operates, right? You can form a team and then you can, really kind of collaborate there very easily. And so is that, is this a similar setup, basically?
This is actually, again, a nice feature of the Galaxy web interface, that you can you can create it's called the Galaxy history. You upload whatever files or you download them from a database directly. You apply some tools, maybe you want to apply ImmuneML, and you have some results, and you can directly share this history with another user, and they can just click the link and continue the analysis where you left off.
So almost like Slack.
Really,
yeah,
it's really a useful feature.
Yeah, that's really useful. That's because that's so, to have that seamless, going back and see what someone else did, and then now you add your value, is really important. I think, where might you see this ImmuneML tool? Here's a question, best used in the type 1 diabetes field. I mean, can you imagine someone would, here we go, sorry. I'm going to unmute him too. There we go. Can you imagine, like, who might come to you, or has anyone come to you from the type 1 diabetes field to use your tool? Not that you know of, nobody yet. Okay, well, we need someone. I think okay, so yeah. Somebody, he's going to, I'm trying to.
You are unmuted, you can speak if you want to.
Yeah.
Yeah, I was just like wondering, I was probably thinking the same that you were thinking, Milena and Lonneke, regarding that. Yeah, I'm actually a bit unsure what we could answer, but I think it's a very relevant question. Let's say it like that. But so I'll try to answer a bit more. Like, I think in general we have. I think in general what we have done in this like interdisciplinary team is that we've had kind of 3 groups, or maybe 4 groups from the start. We've had more like machine learners, which is more like Lonneke, Milena, and me. And then we have had more computational immunologists, which are more like Victor Greif's been involved. And then we've had clinicians involved. And I think there's an interest both from the celiac side locally, but also diabetes. I think we can say that. And then we've had a 4th group that is actually, so the 3 of us need to kind of collaborate in a way, the machine learners, the computational immunologists, and the clinicians. I think that's crucial, and to have that, because us machine learners, we will never kind of know diabetes well enough to be able to like to really do the right interpretation, and there's always a lot in these details. So if we have to kind of summarize our results over to a clinician, we wouldn't maybe pick up on the right details. So I think that's kind of crucial to have these 3 groups.
Yeah, I agree with that totally.
Yeah, and then we actually found out that we need a 4th group, which is actually develop admins, system admins actually. Because in order to make it so what we typically do as machine learners and researchers is to make tools that can be run by other machine learners. But in order to have it being runnable by also clinicians, one needs a bit more sophisticated kind of software solutions than we are typically used to making as researchers. So actually we needed the more technical guys closer to the hardware, and that they're actually sitting there and connecting the wires to the machines. They were actually also very useful for us to help us make it as kind of user-friendly and open as possible for these reproducibility. So they helped us set up these Galaxy system on local servers and make it accessible on web and go through the security and check all of these kind of things. So I found it a bit fascinating that to truly allow collaboration with the clinicians, we also needed to include the most technical people I know of.
Yeah, no, it does. It seems right, because, well, there's also the, you've got like the privacy issue of the data you're handling with clinicians, right? You've got I don't know if you have it over there. I'm sure you do, but we have HIPAA here, and everything has to be anonymized. So who's going to do that? And then, and then it's like, okay, now they also have to, they have to see it through their lens, but wouldn't it be great to have these data in this process seen through the lens of a researcher who actually works, deeply in the immunology, beta cell space. So I think your idea of sort of putting together this dream team is just a great one. And I think that's what it really might take to go to the next level. I think, there's this company called ImmuneAI here that based in Tel Aviv and the Bay Area, San Francisco. And so they have, I think on their target, on one of their targets is, looking at type 1 diabetes, and they use machine learning to look at, a variety of disease states in the immune space. But I mean, how do you compare, contrast what they're doing in a, an industry space versus what you guys are doing in kind of an open science space?
I guess our main focus at Line is actually, as you mentioned, making everything open. So the code is fully publicly available, and we try to make it usable by different user groups. And also, we highly encourage the community contribution. So if there is a group that now wants to develop a specific approach in machine learning that is going to be applied to a problem like this in, for example, disease state prediction, that is kind of something we want to make it so that it's right away available to everyone. And we had a use case like that during the course of development of ImmuneML, where we collaborated with people from the Johannes Kepler University in Linz that developed this DeepRC, which is a deep learning approach to predicting disease status, and that is now also part of ImmuneML. So whatever people would like to develop, we are happy to support and make available to others as well.
Yeah, and that will really serve to accelerate research, I think, in a lot of different places. That's sort of the power of your platform and the power of what you're doing and how you're making it so accessible to other scientists, which is just totally, laudable. And also, I think it really is for the greater good. What disease states have you interfaced with so far?
As Gercheto mentioned briefly, I think the main disease that we've been working on is celiac disease, because there's also leading celiac disease research group here in Oslo. We have a lot of collaborations with them. So there have been, sometimes pilot projects using their data and trying out ImmuneML on their data. So that has been the main one.
That's great. I think it seems to me that you're really poised to interface with the T1D research community. And I would invite, anyone who's listening, excuse me, who is interested to reach out and contact you. Is that something you're up for, or?
Absolutely.
Yeah, I would say we are happy for anyone to put ImmuneML to the test and actually use it on a use case with some disease data.
Fantastic. Well, I'm going to, after this is over, I think I'm going to try to connect you to a couple of people I'm thinking about, mainly at, University of Florida and, the Medical College of University of South Carolina, and places like UCSD, where Kyle Galton is developing or heading up the development of the T1D knowledge portal, which is a big data set that is growing as we speak. So, and those people might be really interested to, to kind of engage in what you have to offer. So I hope that will come to fruition from here. And I mean, I just think, again, what you have developed, as graduate students is phenomenal, and kind of speaks to, your advisor, your PI, as also to your teamwork and your entire extended laboratory and how you are approaching science. And I think it's a fresh and exciting way to, to kind of bring this, make this and then bring it to many different types of researchers and users. So kudos and congratulations on your paper, and keep doing what you're doing. It's phenomenal.
Thank you very much.